I'm here at the beautiful University of Southern California, and I'm so excited to meet Professor Cristina Zavaleta at USC School of Engineering. She is at the forefront of new and exciting technology. She's using nanotechnology to track cancer cells, leading to better treatment that saves lives. What can be better than that?
This is the Zavaleta lab, and we are in the business of actually developing new nano-based contrast agents to detect cancer cells. And in order to detect cancer cells, we need to be able to see where they are, and so we have cancer cells that we use in the lab that have been derived from human patient samples. So they're immortalized human cancer cells. We start things really first in cell culture, and then we slowly move to patients, through, using different sorts of models. But let me show you some of the cancer cells that we are looking at here.
I'm going to be changing the cell medium today for you all. First things first, have to stay clean. Yeah, so this is our incubator that we're going to be dealing with. So, first steps first, we are going to be aspirating out the cell medium. If you can kind of see this pink stuff, that's kind of this serum or this solution that we have that has different kinds of nutrients and proteins that allow the cells to grow. But we're actually going to be replacing it with new cell medium today. So first steps first, we have to get out the old. So now that we have all the old stuff out, we're going to want to put in the new stuff. So yeah, that's just a couple milliliters of cell medium, and now we're going to take it over and let you guys visualize it a little bit. Now we got set up. And as you can see, this is a live view of our cells right now.
Amazing! These are real-life cancer cells that they are using for their experiments.
So we're using our nanoparticles as a way to get them to target onto those cells and then glow.
Nanoparticles are really tiny, even smaller than cancer cells, and we can't even see cancer cells without a microscope.
So the nanoparticles will glow for us in some sort of way, and we're going to show you how they glow next. All right? This is where we are actually going to demonstrate how we're going to make our nanoparticles glow. So there's a bunch of really cool fluorescent dyes that exist that can actually give us signal of light, a light emission. So we have a UV lamp here to help demonstrate how some of these are absorbing light, exciting, and then coming back, relaxing down to the ground state and emitting light. So Ahmed, can you take that and pour a little bit into there, up to about 200 milliliters or so. And this solution is soluble in water, so we'll just give it a little mix. When it's exposed to UV light, and it can turn the switch. It glows pretty crazy, right? Can we turn the lights off just so that we can get even a better appreciation for how bright that is? I mean, it's its own light source, right? Can you imagine surgeons using that as a way to help guide them to where the tumor is? We have several different dyes that we're working with, really pretty colors, and this is just to demonstrate some of the different fluorescent dyes. We have our glowing stuff now. Now we need to get the glowing stuff to the cancer cells, right? Mm-hmm. So the way we do that is we actually form nanoparticles that will carry this stuff with them, and then that will then home to the tumor. And so next we're going to show you some of our nanoparticles that we've prepared so that you can see, you can actually see what they look like. This is Arjun, one of our undergrads, and he is a specialist when it comes to utilizing this instrument. So I told him a little bit about how they we need a way to characterize our nanoparticles. So once we make the nanoparticles and we put that stuff in it, how do we know how many nanoparticles we have and what size the nanoparticles are? Because the size and the concentration are going to affect like how well they get to where they need to go, right? So this is an instrument that does that, and then Arjun will explain a little bit about the Brownian motion that we're seeing from the nanoparticles and how that helps us determine what size it is.
Yeah, so what we have here is basically a sample of our nanoparticles, of our silicon nanoparticles in here right now. And so as you can see on the screen right now, each one of those particles are kind of like moving around a little bit randomly. So what this machine does basically is that we run the sample through the machine at like a certain speed while a laser fires light at it. And so based on the random motion of those particles as they move across the screen, the Brownian motion of those particles, this software is able to track each particle one by one and use that to get a good idea of the size distribution of the particles. And then since it's tracking everything one by one, it can also calculate the concentration of the sample.
We can encapsulate that dye that you saw in there and incorporate it into the silicon nanoparticle so that we can actually see it glow for us. Next, I'm going to go and show you, once we have the nanoparticles and the fluorescent dye, the instruments that we will be using to help determine whether or not they're actually glowing for us. I like to call this like my imaging playground because we have all these different imaging techniques that we use to try and see whether or not our imaging strategies are working. So she's trying to encapsulate that fun stuff that we saw over there into the nanoparticle. Yeah. So now you see that the pellet actually has that color associated with it, right? So all that free dye is off. Now we know that our fluorescent substance is there. So this is the instrument that we use to quantitate how many photons are actually being.
Her name is Amy.
Yes, her name is Amy. Yep. She has a name.
AMI.
It's right there.
So this allows us to like quantitatively compare the dyes that we're investigating. Because sure we can look at something like, oh, this one looks better than this one, but where is the actual numbers associated with that?
We don't have that.
Yeah, we're having it here.
So you can see that each one, like some are brighter than the other.
Yeah, and some are diluted, different degrees. Yeah. But remember, not all fluorophores excite at the same wavelength. That's only UV light. So this machine has the ability to filter out different types of light. So this machine will be able to give us, quantitative assessment of how bright each of the particles are in their respective excitation emission wavelengths.
Do you want to show how to sleep?
Yes.
Which sleep are you guys on?
So you see one of them popping up there.
That's it.
And then these are different emission images. So we're going to sweep across the emission range to determine where they're emitting best at.
They're using this machine they call Amy to test different light on dyed nanoparticles to see which glow the most.
All the machine is doing is projecting light onto the plate and then collecting whatever light it sees based on the filters that we ask it to look for. So fluorescence isn't the only type of imaging modality that we use here. We also use all different sorts of different optical imaging modalities. So another one that we use that has different advantages than fluorescence is something that we call Raman Imaging. Now I'm going to bring you back over here to this room. And now I'm going to pass you over to Alex and Olga to sort of demonstrate how we are using Raman Imaging and what it's providing us in terms of a signal so that we can detect it on cancer. So these are some of the pretty dyes. Each of them has a very unique spectral fingerprint associated with them. And what we're going to show you right now is some of those fingerprints.
So we can just put it on the stage. This is also where we put on our goggles. And we can actually see with this dye that we see some peaks happening here and here and here. And so with the very sensitive optics that we have here, we're able to see these rather fine peaks that are correlated to the molecule structure.
Let's go to the orange one.
Mm-hmm.
And then you'll see it change. So do you know why we use the word fingerprint now?
Yes.
Yeah, because you saw the
So-
difference, right?
yeah.
Between that one and the other. All of them have their
So
own
specific.
unique spectral fingerprints. If you think about the ability to have different batches of nanoparticles, each with its own fingerprint, and then we send each of the nanoparticles to different types of receptors on a cancer cell, it can tell us more specific things about that cancer cell, because maybe this cancer cell expresses 1, 2, and 3, but not 4, but then another cancer cell expresses 1, 2, 3, and 4, and so we're going to be able to get more specificity about that specific cancer. And that's what we call multiplexing, the ability to actually interrogate multiple things at the same time and in one image. Lastly, we have how they're actually interacting in the body. So once we introduce them into a living system, how are the nanoparticles traveling? How long are they in the bloodstream? And we have this system over here in the back. This is our new multi-photon imaging system, and this is Sean and Sebastian, who are both graduate students in the lab. And they're going to tell you a little bit about how we're actually able to visualize the nanoparticles in real time.
So this is real-time imaging of blood flow in a living organism with these green dyed liposomes. It's the same dye you just saw there, flowing through the body. This is actually, I think we were talking about multiplexing earlier with multiple different imaging agents. So we have gold nanoparticles in there, we have G8 liposomes in there. This is just to demonstrate the capabilities of this system. And then also, so this is kind of another way to look at them. So this is separating out the channels. So the red is actually a vasculature label. So it's just a free dye you inject into the body, and then it kind of labels all the insides of the veins. And then you can see the green liposomes really, really starkly against that red background. So this has a vascular label already, but then now watch as the green G8 is injected. And you can see just how bright that G8 is. Just push down on the table. Don't, yeah, on the, yeah. And let go. So that's almost a 1,200 pound table that's lifted up by little air springs. And it keeps the entire system exceedingly stable. Because even there's a road about 500 meters from here, that's enough vibration to shake these kind of images when you're taking them.
Now we're going to do a demonstration to show just how small nanoparticles really are compared to cancer cells. It's called If a Cancer Cell Was the Size of a Donut.
So the donut is the tumor cell, and the sprinkles are the nanoparticles. And so this shows the size difference between them. Our cancer cells in real life are around 10 micrometers, and then our nanoparticles are 100 nanometers. And so what this basically represents is that we have our nanoparticles that have some sort of optical properties, and so when we inject these nanoparticles either intravenously or also they can be taken orally. They will hopefully accumulate in our cancer cell, and then once we image them, we can better outline the margins of our tumor. And so this really helps surgeons in the operating room when they're trying to resect these tumors. And so if they can resect everything, all of the malignant cells, then we don't have to go back and do repeat surgeries. And so better patient outcomes.
I'm going to be talking to some middle schoolers about engineering and different parts of it, but also how there is this kind of inequality in engineering. What would you say to them to encourage them to continue pursuing STEM?
I would encourage them to believe in themselves and to just like I know it's important to have someone advocate for you, right? So I had my mentor advocate and tell me, and I know it's hard to get the confidence to think that you can actually do something that seems really hard, right? In general, people see STEM as more difficult. People get discouraged, but I feel like if you have the right teachers and you have the right people in place, like try and find those advocates to help you. If it wasn't for my mentor in graduate school being my cheerleader, you know, so to speak, I don't know that I would have pursued the PhD track. Like I would have potentially stopped at the master's degree and, you know, not had the opportunities that I have now because I have the PhD, like running my entire own lab. It's pretty special.
That experience was so amazing. Thank you, Professor Zavaleta, for giving us such an in-depth look at your lab and all the amazing work you and your students are doing. I feel so inspired by how STEM can change the world for good, and I hope all you girls out there feel that too. As they say at USC, fight on!